I guess there was some interruption and now it is good to go. So let everyone join. I will start. Let everyone join. I will continue. The people who have joined can write if everything is good to go. Can you see? Can you hear? Everything is good now? Connectivity is good? Please write down something in the chat box, please. Someone from the audience. Thank you, Harsh, for responding. Thank you. Thank you so much. Okay. So let everyone join. In the next few seconds, we are starting. People are still joining now. Okay. Okay. Got it. I got it. So listen, there was a break in the link. Anyways, chalo. I was teaching you culture media. There are 6 type of culture media. I was teaching you the 4th one. I already taught you what is enriched. I already taught you what is enrichment. I already taught you what is selective. We will revise all together in the end. That's why I'm not repeating which one is which now. And 4th one I'm teaching you right now is the indicator. I was teaching you what is indicator, right? I was teaching you what is indicator. Everyone with me? People are still joining. Wait. So I'm teaching you the indicator one, right? So I was teaching you what is indicator medium. So just suppose we are taking a culture medium, and in the culture medium, we are doing an inoculation. For example, here is my inoculum. Hai? So when the colonies are visible, multiple colonies are visible, it is when million times the bacteria have divided, millions of bacteria are there. I want to know before the colony appear whether the bacteria is dividing or not. That is, there is a way to do so. So we have seen, yes, there is a chemical. If we mix that chemical with the culture media, so chemical will change the color. As soon as bacteria divide, the chemical will change the color, and this is known as indicator medium. We have already seen that. We were looking at the 2 examples of the indicator medium. What are the 2 examples of the indicator medium? The first example of the indicator medium is Wilson and Blair. The first is Wilson and Blair and second is McLeod. पहले तो नाम याद करो. First learn the name of the 2 medium. ठीक है? Wilson and Blair is for salmonella and McLeod is for diphtheria. है ना? Learn the corresponding bacteria. Wilson and Blair is for salmonella typhi, typhoid वाला bacteria है, and McLeod is for diphtheria. ठीक है? In both of them, black color is there. Black color. So what is the reason for black color? That you have to learn. What is the chemical which is giving the black color? Name the chemical. In Wilson and Blair, it is sulfide, changes to sulfide, which gives the black color. And in McLeod, it is tellurite, changes to tellurium, which gives the black color. समझ में आई क्या? Everyone got it? Does everyone got it? Please learn. Please learn the 2 type of this thing, indicator medium. Can we proceed? The 5th one is the differential medium. The 5th one is the differential medium. What is differential stain? Differential stain में पढ़ा के आई. What is differential stain? Just a second. Let me open the Telegram also. Yes. So what is differential stain? Differential stain give different color to different bacteria. The same color, the same thing. Differential media give different color colonies to different bacteria. See, the best example is McConkey. I guess everyone know what is McConkey. Tomorrow we will see the detail. Bhai, McConkey क्या होता है? There are 2 type of bacteria: lactose fermenter, non-lactose fermenter. Lactose fermenter form pink colony. Can you see pink colony on the McConkey? And non-lactose fermenter give pale or yellow colony. So different bacteria have different colonies. That is the best example of differential media. McConkey agar is a type of differential media. McConkey is coming in differential. The second example in differential is TCBS. What is TCBS? Don't learn the full form. Just have a look. कोई नहीं पूछेगा full form. है ना? So it is thiosulfate citrate bile salt agar. Thiosulfate citrate bile salt agar. There are 2 type of Vibrio. Vibrio parahemolyticus, Vibrio cholerae. So Vibrio parahemolyticus give green colony and Vibrio cholerae give yellow colony. So you can differentiate the 2 Vibrio by 2 different color. Here we have pink and yellow color. Here we have yellow and green color. So likewise, these are the example of differential media. Different color. Different bacteria give different color. And the last one is the transport media. What is transport media? You know, nowadays home collection is very common. This is home of my patient. My patient is here. Maybe my patient is comatose. My patient is in coma. My patient is unable to move, bedridden. And this is the laboratory. The patient cannot come to the laboratory for giving the sample. So I send someone to collect the sample of the patient here only. So here is the patient sample is collected. The sample can be anything. It can be blood, it can be saliva, it can be CSF, it can be stool, it can be urine, whatever is the sample. I'm expecting it is having some bacteria in that. Okay? So what I assume, the sample should be shifted to the laboratory as soon as possible. And here we will do the testing. Here we will do the testing of that. But it takes 4 to 5 hours, maybe, during the transit time. During the transit time, it takes some hours to ship the bacteria, to transport from the collection to the laboratory. But the problem is that some bacteria are very delicate darlings. They are delicate darlings. They do not withhold the transit time. During the transit time, they just die. They cannot withhold the transit time. During the transit time, they just die because they're very delicate darling. So what to do? So the time where the sample reaches the laboratory, you will get zero bacteria in that. And once you culture, you will get zero growth. And we will give false negative report. Actually, it's not negative. We are giving false negative. We do not want that. So what is the solution of that problem? So solution is that collect the specimen not in simple test tube. Don't collect it in a simple test tube. Collect it in transport media. What is transport media? Transport media will provide a little bit of nutrition to the bacteria so that it do not die. भाई थोड़ा थोड़ा खाना रास्ते भर में खिलाते रहो. बहुत delicate है ना. लंबा खाना नहीं दिया तो मर जाएंगे. तो थोड़ा थोड़ा खाना खिलाते रहो. Give a little bit of nutrition all the way of the transit. Little bit of nutrition is given to the bacteria so that it do not die, so that it reaches the laboratory and then we will culture it. You got it? So this is transport media. Transport media is not required for all bacteria, only for 4 delicate darlings. Learn the name of the 4 delicate darlings. Who are 4 delicate darlings? Number 1, Neisseria. Number 2, Vibrio cholerae. Number 3, Salmonella shigella we take together. And number 4, you can write Streptococcus pyogenes. You can add one. Streptococcus pyogenes. Please learn the 4 name of the delicate bacteria. Neisseria के लिए we require one of the 2 transport media, Ames and Stewart. इन पे PYQ है. Ames and Stewart are the transport media for Neisseria. Learn it. Vibrio cholerae के लिए only one important, Venkataramana Ramakrishnan VR, a South Indian scientist जिसने discovery की है. क्या नाम है? बोलो एक बार नाम. Venkataramana Ramakrishnan VR media या फिर autoclave sea water. This is for Vibrio. Salmonella shigella के लिए buffered glycerol saline या फिर Carrie Blair, right? And Streptococcus pyogenes के लिए Pike's media. You can write down Pike's media. Got it? You have to learn the corresponding pairs. There is no way. है ना? We will revise. So we are done. Everyone with me? I taught you culture media. Can we revise quickly? The 6 type of culture media. You will help me? Will you help me? You have to. So first I taught you enriched. Then I taught you enrichment. Then I taught you selective. Then indicator. Okay, G. Then differential. And last one is transport media. I want the definition of all of them, and more importantly, I want the example of all of them. First, we will try the definition. What is enriched media? It's rich. Rich matlab, it contains nutrient agar plus one of the 3 additional things. Either blood or serum or egg. Okay, that's why enriched media. What is enrichment and selective? We take them together. Enrichment is liquid and selective is solid. Okay, both of them contain either stimulatory substances or inhibitory substances for the bacteria. So that is the thing. Yes, indicator contain a chemical which change color when the bacteria grows. Okay, differential give different color to different bacteria. Naam mein hai. And transport is for transit. For the delicate darlings, not for all. Okay, now you have to tell me the examples in each category. Okay, examples are more important. Enriched mein we have learned 3 examples: blood agar, chocolate agar, and Loeffler's slope. Blood agar contain blood. Chocolate agar also contain heated blood. And Loeffler's slope contains serum. Okay, got it? Hana, please learn. Enriched. Coming on enrichment. Enrichment mein liquid hai. Tell me the broth. So selenite F broth, tetrathionate broth, alkaline peptone water. You have to learn alkaline peptone water, only Vibrio will grow, others will not grow. Tetrathionate broth, only Salmonella will grow, others will not grow. And selenite F broth, only Shigella will grow, others will not grow. You have to learn that. Hai selective media, I'm not telling you here. You have to learn all 30 bacteria. I've given you a list. Tomorrow you read the list. But you get many questions out of that. Okay? Indicator mein again I taught you 2 examples. Wilson and Blair or MacLeod. In both of them, black color is there. Here the reason is sulfite to sulfite. Here the reason is tellurium to tellurite. But the color is black in both of them. Okay, you have to learn the black color and the reason for the black color. Differential में different bacteria have different color. So McConkey में lactose fermenter give pink colony, non-lactose fermenter give yellow colony. है ना? TCBS में Vibrio paramereticus give green colony, Vibrio cholerae give yellow colony. ठीक है? उसके बाद transport is for delicate darlings. You should know the name of the delicate bacteria. Neisseria, Vibrio cholerae, साल्मोनेला एंड शिगेला और स्ट्रेप्टोकोकस पायोजेंस। पहले तो 4 नाम याद करो, डेलिकेट हैं ये, बड़े डेलिकेट, नाजुक नाजुक है ना। सो यू टेल मी, निजीरिया के लिए वी रिक्वायर अमीज एंड स्टुअर्ट, याद करो दोनों, अमीज मीडिया, स्टुअर्ट मीडिया। विब्रियो से तो मुझे एक ही चीज याद आती है, वी फॉर वी, वेंकटरमन रामकृष्णन, वी आर, है ना। शिगिला साल्मोनेला में यू कैन लर्न बफर्ड ग्लिसरॉल, बफर्ड ग्लिसरॉल एंड कैरी ब्लेयर, और स्ट्रेप्टोकोकस पायोजेंस में पाइक्स मीडिया। यू हैव टू लर्न द पेयर्स, देयर इज नो एस्केप। आई एम ट्राइंग टू सिंप्लीफाई, भाई जितना सिंप्लीफाई हो सकता है वी आर डूइंग, लेकिन यू हैव टू लर्न। शिवम, you are asking to explain again differential one. Differential gives different color to different bacteria. Shivam, different color to different bacteria. Can you see? So the details, if you want to know कि मैम ये मैकॉन्की होता क्या है, why lactose fermenter pink बना रही है और non lactose yellow, तो ये I will teach you tomorrow when I will teach you E. coli and lactose fermented non. Just see here, तुम्हें अभी क्या याद करना है कि two different bacteria are giving different colors. You can see the lactose fermenter is giving pink colony and non lactose is giving yellow. Got it? है ना? The same with TCBS. The parahemolyticus is giving green and the cholera is giving yellow. Currently, you have to learn that. Okay, but the detail we will see tomorrow. Okay, Streptococcus का मीडिया है पाइक्स, मुबिन इट्स पाइक्स मीडिया। ठीक है, कल ये पूरा मैं फिर से बताऊंगी, जहां जहां जो जो करस्पॉन्डिंग बैक्टीरिया आएगा, व्हेनेवर आई टीच यू निजेरिया, ये नाम फिर से आएंगे। विब्रियो जब पढ़ाऊंगी वैंट्रम, तो टुमारो यू आर गोइंग टू रिवाइज इट अगेन। आज सिर्फ फाउंडेशन है, कल पूरा रिवीजन है, कल पूरा रिवीजन है स्प्लिटेड फॉर्म में। ओके, कैन वी गो हेड, है ना। ओके, द नेक्स्ट थिंग, लिसन लिसन एवरीवन, ये जो मैंने आपको 6 के 6 मीडियम बताए, कौन कौन से बताए मैंने आपको, इनरिच्ड, इनरिचमेंट, ट्रांसपोर्ट, डिफरेंशियल, इंडिकेटर, व्हाटएवर आई टॉट यू, they all are open, na, they are open like this, it's like a culture plate like this, so it's present in the air environment, oxygen is falling on them, they all are open, oxygen is falling on them. So what are the obligate anaerobic bacteria? What are obligate anaerobic bacteria? Obligate anaerobic bacteria, they are can't breathe air. 3 bacteria, they are strict anaerobe. Can they grow on these? You will say no ma'am, oxygen is present everywhere, they come in contact with the air, they all are open, they are like this in the air, air is everywhere, so air is falling, oxygen is falling, they cannot grow. So what if I grow any of them? I want to grow, can't breathe air, I want to grow them. So how to grow? I cannot grow them on this, I cannot grow on enrichment, I cannot grow them. So I need a special media for them, and that media is known as anaerobic media. Anaerobic media is for anaerobic bacteria that is can't breathe air, in तीनों के लिए. So for the strict anaerobe, obligate anaerobe, we require a special media. The name of that media is anaerobic media. How many of you got it? पहले तो मतलब समझो, anaerobic media अलग से क्यों बनाने की जरूरत पड़ी? Why there is a need to make a separate media that is anaerobic media? Why there is a need at all? जरूरत ही क्या है एक अलग बैक्टीरिया, एक अलग मीडिया बनाने की? Because we want to grow the obligate anaerobes, है ना? There are 2 examples. कौन से 2 examples हैं यहाँ पे? Robertson cooked meat broth number 1 and thioglycolate broth. So first we will see Robertson cooked meat. Thioglycolate, just learn the name. Robertson cooked meat broth. तो कैसे बनाते हैं Robertson cooked meat? इसे RCM कहते हैं. Robertson cooked meat, RCM broth. कैसे बनाते हैं? How do you make it? Take a test tube. ठीक है? Take a test tube. है ना? Please understand how we are making anaerobic. Understand. Fill it with nutrient broth. न्यूट्रिएंट ब्रोथ यानी सिंपल वेज थाली, सिंपल लिक्विड फॉर्म, न्यूट्रिएंट अगर का ही लिक्विड फॉर्म है, न्यूट्रिएंट ब्रोथ, सिंपल न्यूट्रिएंट ब्रोथ, नथिंग, है ना। टेक मीट पीसेस, टेक मीट पीसेस, कहां से आई मीट? मीट इज कमिंग फ्रॉम द ऑक्स हार्ट, ऑक्स का हार्ट. टेक अ ऑक्स, किल द ऑक्स, आई मीन सैक्रिफाइस द ऑक्स, है ना. टेक द हार्ट आउट एंड कट इनटू पीसेस एंड पुट इट हियर. ऑक्स के हार्ट के मीट पीसेस यू हैव पुट एट द बॉटम, है ना. नाउ कवर दिस टेस्ट ट्यूब with paraffin wax, sterile paraffin wax, the mouth of the test tube. Cover the mouth of the test tube with paraffin, liquid not liquid, I mean wax. Wax देखा है, मोम, paraffin, sterile paraffin wax. So that no new oxygen will come. I'm coming on that point, Kavita. Wait, let me finish. I will explain why oxygen. I will come. Listen, no new oxygen is coming here now. No new oxygen can enter. You will say, okay, ma'am, it is completely sealed with sterile wax. No new oxygen can come, we can understand that. But what about the oxygen already present here? There is no vacuum in this world. There is a space, there is oxygen. So what about the space here? There is also oxygen. यहाँ पे तो oxygen है ना. So new oxygen is not coming, we got that. But what about the oxygen already present here? What about that? So don't worry, we will manage that. Heat it on a Bunsen flame. Heat it on a Bunsen flame, है ना. Now whenever you heat it on a Bunsen flame, so ox heart contain some reducing agent inside them. I will tell you the names of that also. Reducing agent that will utilize all the oxygen. That's why ox heart, Kavita, ox heart contain reducing agent. And I will tell you the names also. ठीक है? Ox का heart contain reducing agent that will utilize all the oxygen present in the space. Now, no new oxygen is coming and the oxygen which is already present is utilized. Now it is an anaerobic media. Now it is an anaerobic media and now can't breathe air में से कोई भी bacteria you can grow here. है ना? You want to ask something? You got the answer? So we are taking a test tube. We are filling it with the nutrient broth. We are taking the ox heart meat pieces, putting at the bottom and covering it, completely sealing it. New oxygen cannot enter. The already existing oxygen is utilized by the reducing agent present in the meat pieces, in the ox heart meat pieces. Got it? You should ask me a question I was expecting. Ki, ma'am, how we do the inoculation? Just suppose I want to inoculate Clostridium. Clostridium is can't breathe air ka C. Clostridium को मुझे grow करना है. So if I open it, then again air will enter. So how to do the inoculation? That should be your concern. You should ask me this question. हाँ, तुम्हें पूछना चाहिए था ये क्वेश्चन कि मैम जस्ट सपोज मुझे क्लॉस्ट्रिडियम इनोकुलेट करना है तो कैसे करूं? आई गेस इट इज एनारोबिक, ये तो समझ में आया. बट फॉर इनोकुलेशन इफ यू रिमूव द वैक्स अगेन द एयर विल इंट्रोड्यूस. वी डू नॉट रिमूव द वैक्स. सो विदाउट रिमूविंग द वैक्स हाउ यू डू द इनोकुलेशन? दिस इज माय स्पेसिमेन. जस्ट सपोज दिस इज माय स्पेसिमेन, है ना? आई वांट टू डू द इनोकुलेशन. हाउ आई डू द इनोकुलेशन इनसाइड आरसीएम? Fill your specimen in a syringe with a thin needle, very thin needle, है ना? Put it in a syringe, है ना? And from the syringe make a very minute hole here and put few drops here. This is how we do the inoculation. We do not open the wax. How many of you got it? I'm removing this page. Say yes. Give me a thumbs up. Got it? This is RCM. So RCM contains what? See the image. See the image. The same image I taught you. So this is a test tube. We can see it contains the nutrient broth. Say yes. The bottom contains the meat heart pieces, है ना? Ox heart pieces, and see the mouth. The mouth is covered with paraffin wax. Okay, okay. So the oxygen is not coming, new oxygen, and the already oxygen is utilized. So nutrient broth, है ना? Ox का heart का meat pieces and sterile paraffin wax. This is RCM, Robertson cooked meat. The scientist who created it is Robertson. That's why to honor it, it is Robertson cooked meat. RCM. It is known as. Now you may be thinking, what are the reducing agents present in the ox का heart? So ox के heart में 3 reducing agents are present that utilize the oxygen. Number 1, unsaturated fatty acid. Number 2, glutathione and cysteine. And number 3, sulfhydryl compounds. You don't have to learn, just learn reducing agents are there. You can say that. ठीक है ना? So that is RCM. So we are done with this also. Now we will solve some questions. The first question is in front of you. Who will answer quickly? Who will answer it? The first question is in front of you. Loeffler's medium is it what? Is it an indicator, enrichment, selective, or enriched? I'm waiting for your answer, people. Be fast. Be fast and furious. There is a little bit lag, I guess. Yes, yes. Navjot, absolutely right. Absolutely. So what is the correct answer? You all are right. Very good. The correct answer here is D. It is an example of enriched media. I know. Why does it is enriched? It is not enrichment because it contains serum. It contains serum. I told you 3 examples here. Blood agar, which contain blood. Chocolate agar, which contain heated blood. And Loeffler slope, which contains serum. These all are example of enriched medium. So you get many questions on all 3. If I change the question from Loeffler to blood, answer is same. If I change the question from blood to chocolate, answer is still same. So chocolate, blood, Loeffler, तीनों का answer enriched है. All 3 are PYQ, please learn. Why everyone is not answering? Why only few students, 20, 30 students answer? What about others? Others are sleeping. So please everyone participate. No, it is for your benefit, not me. Okay. The next question is in front of you. Which enrichment media is preferred to grow Vibrio? What is enrichment media? What is enrichment media? Enrichment media is a liquid media. So see, all the options are broth, water. So this word is not helping because all options are liquid. So which is for Vibrio? You tell me that. Which is for Vibrio? Absolutely right. For Vibrio, it's alkaline peptone water. Very good. Alkaline peptone water is for Vibrio. I'm changing the question. Instead of Vibrio, I'm asking Salmonella. What is your answer now? I'm asking Salmonella. Instead of Vibrio, I'm changing it. So what is your answer now? Would you like to change it? You should change it. Now your answer should be tetrathionate broth for Salmonella. And if I change it for Shigella, what is your answer now? For Shigella, it should be selenite broth. So if I change the question, your answer should be changed accordingly. Very good. You all are right. Absolutely right. So this is how we can change the question. The answer will change correspondingly. Okay, Ji. So can we go ahead? So Robertson cooked meat broth is an example of what? Is it enriched media, enrichment media, nutrient agar, anaerobic media? I guess question is very simple. I'm asking for RCM. Question is super duper simple. Yes, the correct answer is anaerobic media. Very good. And now we all know that. The next question. Recommended transport media for Vibrio cholerae. Vibrio cholerae के लिए transport media बताओ. Is it buffered glycerol saline? Is it Venkata Ramana Ramakrishnan VR media? Is it nutrient broth or blood agar? What do you say? I'm asking the transport media for Vibrio cholerae. Absolutely right. You all are right. It is Venkata Raman Ramakrishnan media. You can see VR media is for Vibrio. If I change the question from Vibrio to Salmonella or Shigella, what is your answer now? From the same option, Salmonella or Shigella. In that case, what is your answer now? From the same four options, what do you say? So in that case, your answer will become buffered glycerol. You can see buffered glycerol saline. So you all are right. So in this way, the question can be changed multiple times. Can I come on the next chapter? So I guess this is chapter number 6 or 5 or 6 we are starting since morning. So in this way, we have to complete chapter by chapter. Can I come on the next chapter? It's very easy and small chapter, a little bit. Everyone with me? Can we go ahead? Anna? So let's start the next chapter. The next chapter is antimicrobial resistance. What do you mean by antimicrobial resistance? Usse pehle, let me tell you what is antimicrobial agent. You know, you have studied pharmacology. Pharmacology ki classes ho gayi? Is pharmacology done? The classes are done. You know what are antimicrobial agent? Antimicrobial agent is the agent that kill the microorganism. Let me draw bacteria for you. Anna? This is a bacteria. Okay, just a second. This is a bacteria. This is the nucleus of the bacteria, and this contains the double-stranded DNA of the bacteria. This is the ribosome of the bacteria on which protein synthesis takes place. This is the cell membrane, and this is the cell wall. This is the cell wall. I want to kill this bacteria. Okay, it's okay, it's okay. I'm telling you the classification here. So it will be useful in pharma also. See, so can you see here? This is the bacteria. Being a doctor, I want to kill it because it is present in my patient's body. It is causing a disease in my patient's body. I want to treat my patient, so I want to kill this bacteria. I want to kill this bacteria. There are 4 ways to kill this bacteria. Number 1, I can give a drug that can inhibit the cell wall in this bacteria, and the bacteria will be killed. Bacteria will burst and kill because cell wall will be lost. है ना? So the drugs which inhibit cell wall synthesis, example beta lactams आ जाएंगे, है ना? So there are certain drugs which causes pores in the cell membrane, which causes pores in the cell membrane. So once the cell membrane have the छोटे hole, small hole, the cytoplasm will leak and bacteria will die, है ना? So this is the second way of killing the bacteria. First, give the drug that inhibit the cell wall. Second, give the drug that causes pores in the cell membrane. जैसे कि amphotericin or many drugs are there. ठीक है? Number 3, give the drugs that inhibit the ribosome. That inhibit the ribosome, so they inhibit the protein synthesis. No protein synthesis. Bacteria cannot synthesize protein and bacteria will die. Now, these are of two types. The one which act on 30th component of the ribosome, the one which act on 50th component of the ribosome, the one which act on both. It is of 3 types. The one, they inhibit the ribosome. And the fourth and the last way to kill this bacteria to inhibit the DNA synthesis. Inhibit nucleic acid synthesis, I mean. Inhibit nucleic acid. The bacteria cannot synthesize the nucleic acid and bacteria will die. Inhibit nucleic acid. है ना? These are also of 2 types. Nucleic acid, direct inhibitor and indirect inhibitor. The direct inhibitor are the drugs that directly inhibit DNA synthesis. And indirect inhibitor, they inhibit the folic acid synthesis. Folic acid is required for DNA RNA. No folic acid, no DNA, no RNA. So these are indirect inhibitors. Say yes, these are to kill a bacteria. I'm repeating, what are the 4 ways to kill a bacteria? Can someone help me? Number 1, the drugs which inhibit cell wall, the drugs which cause pore in the cell membrane, the drugs which inhibit protein synthesis by inhibiting the ribosome, either 30S, 50S or both, and the drugs which inhibit nucleic acid synthesis, direct or indirect. People respond. This is from pharmacology. I'm just explaining you कि microbiology में क्या इसका concern है. So can I tell you? So I'm killing a bacteria. I am a doctor. I want to kill this bacteria because it is present in my patient's body. I'm giving the drugs to my patient. The drugs are killing the bacteria in the patient's body. So am I a murderer? I'm doing the murder of this bacteria, no? I am a murderer of this bacteria. Am I? Yes, actually am I. Anna, all doctors are. We are doing the murder of the bacteria. Not only bacteria, of all the microorganisms. Because we want to do the benefit of the patient, not the bacteria. But bacteria is also a living organism, no? You are killing a living organism. Yes, Anna, nobody wants the murder. If someone murder me, I will shout, please help, help, and I will do the defense. I do not want to get killed. The same way bacteria do not want to get killed. Bacteria do not want killing. Nobody wants killing. Nobody wants murder. Of course, है ना? So bacteria produce resistance. So that is my point, है ना? So we want to kill the bacteria, but bacteria do not want to get killed because we are murdering bacteria. Bacteria do not want to do murder. It's not a suicide. It's not by wish. Suicide is by the own wish. Murder is someone else wish. Someone else wish. है ना? So we are killing the bacteria by antimicrobial agent. Bacteria do not want to get killed. So bacteria produce resistance. If someone will murder me, I will do the resistance. No, 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 don't kill me. In the same way, bacteria is doing the resistance. So I am interested. This topic will be dealt in pharmacology आगे का, है ना? But I am teaching you here resistance. The resistance, antimicrobial resistance. It is a resistance to the antimicrobial agent developed by the microorganism. Microorganisms do not want to get killed. Okay, microorganisms do not want to get killed. Got my point? So there are two type of resistance. There are two type of resistance. Mutational drug resistance and transferable drug resistance. Let me explain both. Mutational drug resistance known as MDR and transferable drug resistance known as TDR. I will explain you both. ठीक है? Imagine this is a bacteria. This is the nucleus of the bacteria. This is the DNA of the bacteria. है ना? Now, because of certain mutation, bacteria have developed a gene. This is known as mutational gene. Because of the mutation, this gene have arrived. Now, this gene can be of one. Just suppose this gene is for tetracycline resistance. Now, if I give tetracycline, this bacteria will not die because it has developed a gene inside that protect from tetracycline. Maybe for anything. It is not only for tetracycline. Maybe for cephalosporin. Maybe for penicillin. Maybe any one bacteria. Any one antibiotic. So this bacteria have developed a gene because of mutation. What is the reason? Mutation. म्यूटेशन की वजह से हिंदी में कहूं तो इसने अपने अंदर एक जीन डेवलप कर लिया एक पर्टिकुलर एंटीबायोटिक के लिए, नॉट मल्टीपल. For one particular antibiotic, it can be any antibiotic. Now, this bacteria will not die with that antibiotic. है ना? So, if the gene is for tetracycline, it will not die with tetracycline. If the gene is for penicillin, it will not die for penicillin. Say yes. Got it? Now, this bacteria will divide into two daughter cells. All bacteria divide every 20 minutes. So, this is also dividing. So, it will not die, but it will give the gene to the progeny also. It will give the gene to the daughter cell also. And they will pass the gene to their daughter cell. I mean, the mutated gene once occur. यहाँ तो पहली बार हुआ. Here mutation is first time, not is transferred generation to generation to generation to generation. This is known as vertical drug resistance. Vertical से मेरा मतलब है generation to generation, from one generation to another. So the parent is not dying with tetracycline. The daughters are also not dying. Their daughters are also not dying. The progeny is also not dying. It is known as vertical resistance. Say yes. The mutational one is vertical. The mutational one is resistance is vertical resistance. It is generation to generation. Say yes. It is for one antibiotic at a time, not multiple, one at a time. One at a time. And it is low degree. Low degree. The bacteria is not killing with penicillin, no. But if you increase the dose of penicillin, it will die. It is of low degree resistance. बहुत ज़्यादा नहीं है. One at a time. है ना? तो ये हुआ आपका mutational वाला. This one is mutational. It is vertical. One by one. I mean generation to generation. Can you see this diagram? सिर्फ ये वाला देखो. Mutational wala. Don't see transferable. See only mutational one. Can you see multiple bacteria in this petri dish? है ना? You put tetracycline. They all will die. But one of them, can you see color change? One of them, this one is not dying. This one. You put tetracycline. This one is not dying. Rest all will die. Rest all. Because it is having the gene for tetracycline. It is not dying with tetracycline. Now it is producing progeny and giving the same gene to all the progeny. So all the progeny will not die. So it is generation to generation. You got it? Anna, you got it? Okay. Yes, one drug at a time. But see, there are two friend bacteria. This is a friend, this is a friend. Two best friends are there. ठीक है? Friend A, friend B. Name them. ठीक है? Friend A have a plasmid. है ना? For some particular drug. Friend B don't have that. ठीक है? Just suppose the plasmid have multiple drug resistance. For tetracycline also, cephalosporin also, penicillin also. So if you put any of the drug, this bacteria will not die. But if you put any of the drug, this will die. A have resistance for multiple, B don't have. Plasmid में multiple होता है, not one, multiple resistance at a time. है ना? Tetracycline B, cephalosporin B, penicillin B, तुम कुछ भी डाल लो, A नहीं मरेगा, लेकिन B तो मर जाएगा. अब B बोल रहा है मैं तो तेरा best friend हूँ यार, है ना? B saying to A, I am your best friend. No, it is not good thing when tetracycline will come, you will survive, I will die. Do you feel good? तेरे को अच्छा लगेगा क्या मैं मर जाऊंगा तो? So you give the gene to me also. मुझे भी दे वो gene. So B, A is saying, okay, let's come together. Let's do sexual intercourse. So sex in bacteria. Sex in bacteria is conjugation. This is known as sexduction. है ना? So the two bacteria are coming physically closer. They are making a tube between them. They are making a tube between them. By that tube, diagram is wrong little bit. You can correct. By that tube, bacteria A is duplicating the plasmid. One copy is kept by himself and one is given to the partner. Now when tetracycline, penicillin, anything will come, both will survive. So it is given through the same generation, not the next generation. The resistance is transferred in the same generation. It is known as horizontal transfer. Say, so this one is vertical, this one is horizontal. इसको transferable क्यों कहते हैं? Because it is transferred via plasmid from one bacteria to another. और इसको mutational क्यों कहते हैं? Because it is via mutation. First एक bacteria में mutation and then generation to generation. और ये transferred from one bacteria to another. So we have studied two type of drug resistance: mutational drug resistance, transferable drug resistance. Mutational is first time mutation होगा and then transferred from generation to generation to generation. It is known as vertical transfer. Transferable drug resistance is through plasmid. Plasmid means sex in bacteria, conjugation, and it is क्या कहते हैं, horizontal drug resistance between the two same generation. Horizontal. कितनों को समझ में आया? हाँ? It is one drug at a time. It is for multiple drug. It is low degree. It is high degree resistance. Say people respond. Either say yes or say to repeat. Say something but respond. Don't sit like this. Not it. Say something. Okay, so there are two type of drug resistance, huh? So this is the two type of drug resistance in front of you. You can read. Okay, so mutational drug resistance is there and transferable drug resistance, MDR, TDR, MDR and TDR. So Anita, learn the summary. There are two type of drug resistance. एक होता है MDR, एक होता है TDR, है ना? MDR generation to generation जाता है. ये parent to progeny to progeny to progeny. That's why MDR is known as vertical. वर्टिकल मतलब ऐसा लाइन, है ना? फर्स्ट टाइम म्यूटेशन हुआ और उसके बाद जनरेशन टू जनरेशन टू जनरेशन टू जनरेशन टू जनरेशन. टीडीआर इज बिटवीन टू फ्रेंड बैक्टीरिया. It is the same generation, not different, same generation. So this one is known as horizontal. See, the line is horizontal here. So this is vertical, this is horizontal. ठीक है? This is through plasmid. This one is through plasmid. I will explain you the plasmid. अभी आएगा genetics में. But just learn, है ना? Okay. No, Shivam, in TDR it will not go to the progeny. है ना शिवम? in TDR, in MDR it will go to the progeny, in TDR it will not go to the progeny. Shivam, the answer is that because TDR is through plasmid, MDR तो DNA में हो रहा है ना, the mutation is in the DNA, so DNA get transmitted, but plasmid not necessarily transmitted to the progeny. You got your answer? Huh? So that is the thing. TDR is don't get transferred to the progeny. It is through the same generation. The two friend bacteria can be there. ठीक है? Here one drug at a time and here multiple drug at one time. ठीक है? And this is of low degree, this is of high degree. That's all. You have to learn that. Okay जी, है ना? This is one drug at a time, this is multiple drug at a time. This is low degree, this is high degree. ठीक है? Here the resistance is transferred from offsprings, one generation to another generation vertically. And here it is horizontally by conjugation sex in the bacteria between two friend bacteria. I tried my best. Okay? So the two type of the drug resistance. You will get questions from this topic, but in pharmacology. In the pharma also the same topic is repeated. There you will get a better understanding, but you have the overview here. Okay? So to change your priorities, to change your life, you have to change your priorities. Decide what is your target of your life, what you want in the next few months, in the next few years, what you want to see yourself. Accordingly decide your priorities and follow your priorities. Priorities changes with time. It is not always the same priorities. Okay, so phasic hai wo. So is phase mein what is your priority? Just decide that. Anyways, coming to the next topic, bacterial pathogenicity. Okay, here the most important topic which I would like to tell you, the two type of the toxin, endotoxin, exotoxin. There are two type of toxins in the bacteria. You get many questions on that. देखो, नाम सुना है? I guess everyone have heard the name. There are two type of toxin. एक होता है exotoxin, एक होता है endotoxin. Exotoxin, endotoxin, दो तरह का टॉक्सिन होता है. देखो, I will explain everything of that. Exo को exo क्यों कहते हैं? Why exo is known as exotoxin? देखो, this is the bacteria. This is the cytoplasm of the bacteria. The toxin is secreted out. The toxin is secreted out of the body of the bacteria. That is exotoxin. Secreted out. Exo मतलब बाहर, है ना? That's why exotoxin. It is a live bacteria. The live bacteria is secreting the toxin out. What is endotoxin? Okay. Before that, one more thing I forgot to tell you. Exotoxin seen in gram positive bacteria, endotoxin seen in gram negative. You remember the cell wall of the gram negative? बताई थी मैंने, एक रियल मेम्ब्रेन, एक आउटर मेम्ब्रेन, वन मोर मेम्ब्रेन जिसे आउटर मेम्ब्रेन कहते हैं. दोनों के बीच में द स्पेस इज नोन एज पेरिप्लाज्मिक स्पेस, और उसमें होता है लिपोपॉलीसेक्राइड. याद आया क्या? द लिपोपॉलीसेक्राइड जो मैंने बोला था ना, दैट इज एंडोटॉक्सिन. The lipopolysaccharide is endotoxin. Actually, it is a component of the cell wall. It is not toxin, it is a component of the cell wall. It is not a toxin. I'm repeating, it is a component of the cell wall. So whenever the bacteria burst, at that time it comes out. So it is secreted, it is coming out of the killed bacteria. Here the bacteria is live, here the bacteria is killed. Here toxin is synthesized in the cytoplasm and secreted out. Here toxin is not synthesized. The toxin is a component of the cell wall. Can you see? So whenever the bacteria is bursting, it is coming out. Say yes. Live bacteria, dead bacteria. Here toxin is coming out. Here it is a component of the cell wall. When the bacteria burst, the cell wall का fragments में वो बाहर आएगी, है ना? This is gram positive bacteria. This is gram negative bacteria. इतना समझ में आया? इतना समझ में आया? Now there are 4 things which are more in exo and less in endo. Number 1, exotoxin are highly potent. They are less potent. ठीक है? They are highly antigenic. They are less antigenic. Antigenicity मतलब क्या होता है? Whenever both enter in my body will make more antibodies for exo but not many antibodies for endo. That is the meaning of antigenicity. है ना? They are highly potent, highly antigenic, highly specific. They are less specific. And they are highly enzymatic. They are less enzymatic. Learn the 4 properties. है ना? Potency, antigenicity, specificity और enzymatic. All the things are more in exo as compared to endo. समझ में आई क्या बात? They all are more in exo as compared to endo. Everyone got it? लेकिन इनमें 4 अच्छी चीजें हैं तो एक बुरी भी है. एक बुरी चीज है. 4 more है, लेकिन this is heat labile. Once you heat it, they get destroyed. 4 strong है, ये highly potent है, highly antigenic है, highly specific, highly enzymatic. लेकिन गर्म करो तो फुस, है ना? They just get destroyed. ये सब कम है. They are less potent, less antigenic, less enzymatic. लेकिन these are heat stable. गर्म करो तो they are stable. They will not get destroyed. So these are all the properties. The last thing, composition. इनका composition क्या है? हाँ? So जो exotoxin है, they are protein, polypeptide. They are polypeptide in nature. These one are protein, polypeptide. खाने में क्या 3 चीजें खाते हो? एक तो प्रोटीन खाते हैं. We all eat protein. What other things we eat? Carbs and lipid. So this one is protein. The other one is the carb and lipid. Carb and lipid means lipopolysaccharide. Lipid and polysaccharide. So one is protein and one is carb and lipid. समझ में आ रही है? This is how you can learn. So this is polypeptide. This is lipopolysaccharide. So please help me in learning the differences between exo and endo. I tried my best. Exo, endo. Okay, no. I will ask you. You have to tell me. I will not tell. So you have to tell me. Exotoxin and endotoxin. Okay. Can you please explain specific and sensitive ones? These 2 terms confused. So Kavita, these are the 2 terms in the PSM you will learn, but I will explain you when I will come on the test. So these are related to the test, some test we have to do, na? Specificity and sensitivity. Anyways, chalo. Exotoxin, endotoxin. Listen. Exotoxin and endotoxin. First, you have to tell me the bacteria. Which bacteria secrete which one? Then you have to tell me which one is secreted by live and which one comes out of the killed bacteria. Then you have to tell me the mechanism. कैसे आते हैं बाहर? Then you have to tell me regarding the potency, antigenicity, specificity, and enzymatic action. ये चारों किसमें ज्यादा हैं, किसमें कम है? Okay. Then you have to tell me the composition, heat relation with the heat, and you have to tell me the composition also of each of them. So let's start it. People start speaking. Please start speaking. Tell me the bacteria. Exotoxin is secreted by gram positive, endo by gram negative. Please learn it by heart. Now you tell me the live or dead. So this is secreted by live bacteria. This is by killed bacteria. है ना? Tell me the mechanism. The mechanism, this is secreted out. And this is a component of the cell wall. This is not secreted out. When the bacteria get bursted, it will come out. है ना? Now tell me the composition of both of them. So this one is protein, that is polypeptide. And this one is lipid and polysaccharide, lipopolysaccharide. So this one is polypeptide, this one is lipopolysaccharide. है ना? Now the 4 things. Potency, antigenicity, specificity, and enzymity. All the 4 things are more here. All the 4 things are less here. Please learn. 4 things more here. Lekin. There is a twist. 4 things are more, but this is heat labile. जरा सा गरम करो फुस हो जाएगी. And this is चारों thing कम है, लेकिन this is heat stable. एक चीज अच्छी है. चार अच्छी हैं तो एक बुरी है, चार बुरी हैं तो एक अच्छी है. So please learn like that. You will never forget and you get many questions on this concept. Say yes. Everyone say yes. Everyone got it? Everyone got it? Huh? So this is the thing. So same table in front of you. Now you can read the table. No need to read the table. You already got it now. So learn. This is exotoxin. This is endotoxin. Learn. This is gram positive. This is gram negative. This one is polypeptide. This one is lipopolysaccharide. This one is secreted out by the living cell. This one by the killed cell. Integral part of the cell wall on disruption of the cell. ठीक है. This is heat labile. This is heat stable. अब चार चीजें है ना. So enzymatic action, specificity, potency, है ना. These all are more here. These all are less here. So this is how you have to learn the differences. ठीक है. Now after that, the next topic. This you read in PSM also. What is endemic disease? What is endemic disease? What is epidemic and what is pandemic? Can you see the three type of diseases? Endemic, epidemic and pandemic. Can you see this is globe? ये पूरा अर्थ है. You can see this is a globe. This is a globe. This is a globe. ये पूरा का पूरा अर्थ है. Globe है ना? So here endemic, the disease is constantly present but in low burden. Let me explain. How many children, how many students are there in class? Suppose 100 students are attending my class now, है ना? So 100 में से 5 of them have flu, है ना? सर्दी खांसी जुकाम है. ये 5 को ठीक होगा तो अगले 5 को हो जाएगा. These 5 will get cured, अगले 5 को हो जाएगा. So the disease is endemic. Endemic means constantly present in a population. It is constantly present. Some get cured, others will have. Those will get cured, others will have. But it is in low burden. It is not a public health problem. It is in low burden. The burden is low. Say yes, the burden is low. समझ आई क्या बात? The burden is low. So disease is endemic but burden is low. ठीक है? Epidemic का मतलब क्या है? Sudden clustering of cases at one region, है ना? 100 में से अगर 60 people को एक साथ फ्लू हो गया, तो I will say the disease is epidemic. Sudden clustering of cases. और pandemic means crossing the continents. Continents cross करके involving whole globe, है ना? So you know these terms, I guess. So read the question, tell me the answer, quick, fast. Read the question, tell me the answer, quick and fast. Huh? Which of the following statement is not true for exotoxin? Not true for exotoxin. Read it. Slowly read it and then tell me. Exotoxin के लिए क्या true नहीं है? They are protein, say yes or no? Huh? Say yes, ma'am. They are polypeptide only. They are highly antigenic. Yes, highly antigenic, highly specific, highly enzymatic. We know. They are heat labile. हाँ, चार चीजें अच्छी हैं लेकिन एक बुरी है. Heat labile होता है. What you people are saying AAC? क्या आंसर बोल रहे हो यार? I am saying which is not true. मुझे गलत बताओ exotoxin के लिए. They are obtained by rupture of cell. No, they never obtained by rupture of cell. Rupture of cell से तो endo आता है. So correct is D. The people who are saying A, B, C as answer, have you realized your mistake? The correct answer is D. काफी सारे बच्चे A और C भी बोल रहे थे. You realized? कोई बात नहीं. You realized it? हाँ, बस the thing is you should realize कि कहाँ गलत थे. ठीक है? The correct answer is D. The correct answer is D. Only D is the answer. Everyone got it? Can we go ahead? So you get many question on exo and endo. मैंने तो सिर्फ एक डाला है prototype, but you get many question on this concept. ये difference की table at the tip of your tongue. The next question is in front of you. There is a health center. There is a health center reports 40 to 50 cases in a week. हर week कितने cases आते हैं? 40 to 50 एक particular disease के. This week 48 cases. So you tell me, is it epidemic, sporadic, endemic or outbreak? Every week 40 to 50, but this week 48. So 48 in the same range. 40 to 50 हर हफ्ते आते हैं. इस हफ्ते भी तो उतने ही हैं. It is 40 to 50 only. So it is constant present. So I will say it is endemic. It is endemic. Instead of 48, अगर मैं यहाँ पे आपको 200 cases दे देती, तो it will become epidemic in that case. So currently it's endemic only. Got it? So next chapter we are coming is genetics. है ना? So let me take the genetics after which we can take the break. है ना? 2 chapters are still remaining in general. एक तो genetics है और एक sterilization. है ना? Sterilization. Genetics and sterilization है. That is after that we have complete immuno. In immuno also we have 8 or 9 chapters. They are very small chapters. लेकिन हमें वो complete करना है ना? So target is before lunch we complete general. After lunch we can take immuno, but it's okay. Immuno थोड़ा छोटा है तो भी 1 chapter overlay चलेगा. So okay. Sporadic. Namrata is asking sporadic. Sporadic means if the 2 cases are there, Namrata, they are not related to each other. neither by time nor by space. Just suppose एक case America में हुआ, एक India में हुआ. The two cases not never come in contact with each other. So they are sporadic, sporadically अलग-अलग हुए दोनों. They are not related with space with each other. They never come in contact and not in time also. So that is the meaning of the sporadic. Got it? So can we start the genetics now? Are you people with me? It's a small chapter yet very important and many questions come from the genetics. So can I start genetics? Let's start genetics, bacterial genetics. You tell me how many chromosomes are present in human. This is a human cell. How many chromosomes humans have? This is the nucleus of the human. You will say, ma'am, humans have 46 chromosomes. Or you can say 23 pairs of chromosome. Either say 46 chromosome or 23 pairs of chromosome. Pair number 1, 2, 3, 4, 5. Likewise, we have 23 pairs. What about the bacteria? What about the bacteria? How many chromosomes are present in bacteria? This is a bacteria. Bacteria don't have nucleus. Bacteria have only one chromosome, that too circular. One chromosome, only one chromosome, that too circular, not linear, one chromosome. Humans have 46, bacteria have 1. Humans have linear, bacteria have circular. Okay, so this is a circular. So let me draw the bacterial cell. Let me draw the DNA. Where is the DNA? This is the nucleic acid. One circular chromosome. In the bacteria, we have only one chromosome. That too circular, it's in front of you. Let me ask you a question. Can you tell me extra chromosomal genetic material? Extra chromosomal genetic material in the bacteria. Can you tell me extra chromosomal genetic material in the bacteria? Where is extra chromosomal? What do you mean by that? Does the bacteria have DNA apart from chromosome? ये chromosome के अलावा कहीं DNA है क्या? Chromosome में तो DNA है. We know chromosome में DNA है. Chromosome में genetic material है, DNA है. We all know that. Apart from chromosome, does it have DNA anywhere else? Yes, at two places. So where is that? Where is that? It can be present in the cytoplasm. The DNA can be present in the cytoplasm. It is known as plasmid. So plasmid is one of the extra chromosomal genetic material. Number one. Sometimes this plasmid go and fuses in the nucleus. Plasmid is present in cytoplasm. It is in cytoplasm. Sometimes it go and fuses in the nucleus. It fuses in the nucleus. And this one is known as episome. This one is known as episome. It is present in nucleus. So plasmid and episome both are extra chromosomal genetic material. समझ में आई क्या? See the two diagrams. See the two cells in front of you. Can you see the two bacteria? See, in the two bacteria, this is the chromosome. Let me show you the chromosome. So this is a chromosome present in both of them. This is a chromosome here and this is a chromosome here. Everyone got it? Chromosome तो दिख रहा है. है ना? Apart from chromosome, where is the genetic material? If it is present in the cytoplasm, it's plasmid. It can be multiple. When the plasmids are coming and fusing here, see, they are fusing here. So same plasmid is known as episome. Everyone got it? Plasmid को ही episome कहते हैं. So plasmid is present in the cytoplasm, episome present in the nucleus. Say yes. Everyone respond. Say yes. Got it? Now let me tell you the ways in which the genetic material, the gene get transferred in the bacteria. Can we transfer the gene from one bacteria to another? There are 4 ways. Very important, ultra important topic. Make 5-star over it. Please, it's a ultra super duper important topic. So there are 4 ways in which the gene get transferred from one bacteria to another. What are the 4 ways? Transformation, transduction, lysogenic conversion, and conjugation. These are the 4 ways by which the gene get transferred from one bacteria to another. Okay, so let me tell you. Let me draw bacteria here. Okay, on one page only I will teach you all 4. This is a bacteria. This is a bacteria. See the 2 bacteria. Everyone. This is the DNA of one bacteria, red color. This is the DNA of another bacteria, blue color. ठीक है? Now let me tell you the 4 ways. The first is transformation. देखो everyone on the screen. Imagine the first bacteria is burst. The first bacteria get bursted. Yes, it get dead. The first bacteria is killed. The first bacteria is dead. है ना? So what will happen? When the bacteria is dead, it will burst. So the DNA will leak out. DNA from the dead bacteria will leak out and this free DNA is entering in the second bacteria. We call it a free or naked DNA. This is free or naked DNA which is entering in the second bacteria. Now after entering in second bacteria, there are 2 possibilities. Either it will make a plasmid or it will go and integrate and form the episome. It's its choice. But this method is known as transformation. Transformation में कोई vehicle नहीं चाहिए, है ना? DNA खुद पैदल जाएगा. You got my point? So one bacteria is always dead. DNA will come out, leak out, and DNA will go itself to the second bacteria, and this is known as transformation. You have to learn the word free or naked. No vehicle is required. How many of you got it? How many of you got it? So the first way I already taught you, transformation. Say yes, everyone, say yes. So you can see the same diagram, transformation. First bacteria is not shown to you, it's dead. See the DNA coming out of the first bacteria. This is the DNA coming out of the first bacteria, entering the second bacteria. After entering the second bacteria, it is entering freely. It is entering freely. है After entering, either it is forming a plasmid or it is forming a episome. This is known as transformation. है ना? मैं कुछ भी बोलूंगी तुम कुछ भी मानोगे, ऐसा तो नहीं होगा, है ना? You will ask a proof. मैम, is there is a proof of transformation? ये सच में होता है? Proof क्या है? What is a proof? Can we do a proof? So Griffith experiment. Please learn. Griffith नाम का scientist है जिसने एक experiment किया. The name of the scientist is Griffith. The Griffith has done an experiment to prove transformation. Griffith has proven the transformation with the help of an experiment. Say yes. है ना? So let me explain you Griffith experiment. Griffith has taken 4 चूहे, 4 mice. How many of you understand Hindi? Or rather, how many of you don't understand Hindi? चलो, doesn't have a concise in English only. Take 4 mice. है ना? 4 चूहे ले लो. 1, 2, 3. अच्छा, before starting that, let me tell you, I am explaining you Griffith experiment, है ना? Griffith experiment to prove transformation. पहली बात, transformation को prove करने के लिए कर रहे हैं हम Griffith experiment. ठीक है? We will take 4 mice later on. लेकिन first understand there is a bacteria. The name of the bacteria is Streptococcus pneumoniae. Have you heard about this bacteria? Streptococcus pneumoniae. Pneumococcus भी कहते हैं इसको. Pneumococcus. सुना है? Pneumococcus नाम का एक bacteria होता है. Tomorrow I will teach you all bacteria. I will teach you this also. Pneumococcus. It is of 2 types. Let me draw the 2 types. See, this is a bacteria. This is a bacteria. This one have the capsule. This one don't have the capsule. So this is the 2 type of pneumococcus. You will see, ma'am, this one is capsulated. This one is non-capsulated. दिख रहा है वो तो diagram में. I got it. Capsule की वजह से ना वो smooth हो जाएगा. So this one is known as smooth form. Smooth form also known as S form. और बिना capsule it is rough. ये उबड़ खाबड़ है पूरा, rough है. So इसे कहते हैं rough form. इसे क्या कहते हैं? rough form या R form. क्या कहते हैं? rough form या R form. समझ में आया? So there are 2 types of pneumococcus. The capsulated is the smooth. Learn करो, capsule की वजह से smoothness आ गई. और बिना capsule it is rough. It is rough. So rough, है ना? Capsule is required for causing disease. So this one is virulent. This one is pathogenic. This one is virulent. It causes the disease. है ना? ये pneumonia कराएगा. Pneumococcus pneumonia कराएगा. And this one is avirulent. It do not cause the disease. है ना? It is like a common cell. Body में जाएगा भी तो कुछ नहीं कराएगा. It will not cause anything. You got it? Let me take 4 mice. Now you will tell me the result. Everyone on the screen. देखो मैं पूछूंगी. You have to tell me the result. Mice number 1, mice number 2, mice number 3, mice number 4. In the first mice, I am injecting live S. तुम बताओ क्या होगा? सोच के बोलना. In the second, I am injecting live R. तुम बताओ क्या होगा? In the third, I am injecting killed S. है ना? Fourth में मैं बाद में बताती हूँ. First, you tell me the result. हाँ, first mice में क्या होगा? You will say, ma'am, S. S क्या करता है? S तो virulent होता है, capsulated होता है, उसके पास तो virulence है. तो this mice को हो जाएगा pneumonia and this mice will be dead because of the pneumonia. Because it is live S. S is virulent. I told you. है ना? Not only this. If you take the blood of this mice, you will get what you have injected. You have injected live S now. You get live S. You will say, ठीक है ma'am, whatever we injected, we got that. Just a second. Whatever we injected, we got that. Make a sense. है ना? जो डाला वो मिला. ये तो समझ में आ रहा है. ठीक है. Second का result बताओ. You injected live R. Does R causes disease? You will say, no ma'am. R is rough. It is non-capsulated. It is avirulent. This, if you are injecting this to the mice will be healthy. Mice को कुछ नहीं होगा because it is non-capsulated, है ना? So the mice will be healthy, and if you are taking the blood, whatever you have injected, same you will get. You have injected live R, you get live R. You will say, ma'am, make a sense, है ना? This also have a sense. ठीक है, इतना भी समझ में आया. Nothing rocket science. जो सोचा है वही समझ में आया. तीसरा भी ठीक है. चलो तीसरे पे आओ. You have injected killed S. इसमें क्या होगा? तुम बताओ क्या होगा? S क्या होता है? आप बोलोगे मैम S तो virulent होता है. S is virulent, but कभी killed bacteria disease कराता है? Can killed bacteria cause a disease? Killed bacteria can never cause a disease. कितना ही virulent कितना हो, howsoever virulent it is. But if the bacteria is killed, howso it can cause a disease? It will not cause any disease. Still, this mice is also healthy. This mice is also healthy. है ना? And if you take the blood again, whatever you have injected, you get the same thing. Till now, the story was, everything was making a sense and we can get it. Now tell me, I'm not interested in mice number 1. I'm interested in mice number 2 and 3. Tell me the result. 2 result is healthy. 3 result is healthy. I have combined 2 and 3 and injected in 4th. I have combined 2 and 3. Live R and killed S. Okay, I have combined the 2. Live R cannot cause the disease because they are R. R cannot cause the disease. And killed S also cannot cause the disease because it is killed. Killed bacteria cannot cause the disease. So I have injected. So when alone live R is given, the mice was healthy. When alone killed S was given, the mice was healthy. So I was expecting that mice are healthy ho, but an eye opener, a shocking experiment, the mice was dead. Can anyone explain the reason for mice is dead? Why the mice is dead? Why the mice is dead here? Neither live R can kill the mice, nor killed S can cause disease in the mice. तो mice में dead हुआ कैसे? Or not only this, if I took the blood ना, I get live S. Oh my God, live S तो डाला ही नहीं था. I have injected live R and killed S. Live S कहाँ से आ गया? Live S कहाँ से आया? Can I? Yes, very good. Yes, Sukhchin, everyone who is answering. Mice के अंदर transformation हुआ. The fourth mice have the transformation. Can you see? The killed S का DNA leak out. Killed bacteria ना rupture हो गया वो. The DNA leak out. The DNA leak out and the free DNA go in the live R. The free DNA go in the live R. DNA जा रहा है ना? It is carrying the gene of the capsule. It is carrying the gene of the capsule and live R started making capsule and it become live S. Live R converted to live S. Transformation take place and this is proven transformation actually takes place. This is Griffith experiment. How many of you got it? Give me a thumbs up. All live students. I mean all live who got it. And they are live also and they are alive also and they have understood also. हाँ? जिंदा हो, देख रहे हो, जाग रहे हो, समझ में आई बात? कोई confusion है इसमें? So this is Griffith experiment. So Griffith experiment with दो क्वेश्चन आते हैं. Number one, Griffith experiment is for proving what? Is it for proving transformation, transduction, conjugation? क्या prove कर रहे हैं हम इससे? Transformation. Griffith experiment में बैक्टीरिया कौन सा लेते हैं? So we take a pneumococcus, Streptococcus pneumoniae, the S form and R form, and you can understand कि कितने क्वेश्चन इसमें से बन सकते हैं. They can give you the different scenario. ये डाला तो क्या, ये डाला तो क्या, ये डाला तो क्या, ये डाला तो क्या. So if you have the concept, you can make it out. ठीक है? Got it? That is transformation. So we are done with the first. There are 4 ways of gene transfer. Gene transfer के 4 ways हैं. Okay, you people are too much. Okay, listen. There are 4 ways of gene transfer. Transformation is done. ठीक है? Now I'm teaching you transduction, lysogenic conversion, and conjugation one by one. So coming on the second one, transduction. Transformation is done. Transduction. ठीक है जी? So this is a bacteria. This is a bacteria. ठीक है? This bacteria have the DNA red color. This bacteria have the DNA blue color. So there is a third thing. There is an agency. It is known as virus. The virus is known as bacteriophage. This is bacteriophage, the virus. It is a virus. So what this virus is coming in the first bacteria, इसमें से stealing a little bit of DNA of this. इससे चुरा के आ गया थोड़ा सा DNA. हिंदी में कहूँ तो it is stealing a little bit of DNA of this. This is known as packaging error. And after coming out, it is going to the second bacteria. Whatever taken from there, it is given to the second bacteria. है ना? So there is an agency, a vehicle. In transformation, DNA was going itself, walking. Here it requires a car. Who is the vehicle? Bacteriophage. So this method is known as transduction. In transduction, the agency is bacteriophage. Bacteriophage is doing pick and drop service. Pick service, drop service. Pick from one place, drop at other place. Do you have Swiggy at your city? Zomato, Swiggy, pick and drop. है ना? They just pick the item, they just drop the item. So they are just Zomato, Swiggy like. है ना? They are picking, they are dropping. So this is the agency. This is known as transduction. How many of you got it? This is transduction. You got it? You can see the same. It is through a bacteriophage. है ना? So from one bacteria to another bacteria, the agency is bacteriophage through a packaging error. So same diagram is there. Anyone have any confusion? This is transduction. The third one is confusing. Lysogenic conversion. The third one is lysogenic conversion. See, I'm drawing the second and third together. So first, let me draw what is transduction. There are the 2 bacteria. In lysogenic conversion, we have only 1 bacteria. We don't have 2 bacteria. Here we have the DNA. है ना? In lysogenic conversion, in transformation, I told you there is a bacteriophage. The bacteriophage is taking from here and dropping here. It's pick and drop. So a little bit is taken from one bacteria and dropped to the other bacteria. So it is pick and drop service and it is through bacteriophage. I guess everyone got it. ये तो सबको समझ में आया. This is transduction. ये तो सबको समझ में आया. Now I'm teaching you lysogenic conversion, which is confusing. Lysogenic conversion. Here also we have a bacteriophage. That's why I'm drawing together. Here also we have a bacteriophage. This is green DNA of the bacteriophage. So we don't have other bacteria. Bacteriophage is giving own DNA to the bacteria. It is not pick and drop. Bacteriophage अपना DNA bacteria को दे रहा है integrate करने के लिए. This is lysogenic conversion. How many of you got the difference between transduction and lysogenic conversion? How many of you got the difference? In both of them, we have a bacteriophage. Here, bacteriophage is doing pick and drop. Here, bacteriophage is contributing its own genetic material. People respond. It's difficult, but I'm trying hard to explain you. I know. So please understand. Ultimately, we are doing genetic material transfer in the bacteria. I know. So here it is between two bacteria, here between bacteria and virus. Got it? I tried hard. This is lysogenic conversion. Now read it. Lysogenic conversion. What does it mean? Yes, खुद का दे रहा है सद्दाम. Absolutely right. Own DNA. है ना? Phage own DNA is integrated with the bacterial chromosome. Own DNA. है ना? So own DNA दे रहा है वो. So that is lysogenic conversion. Got it? So we got the difference between the two. What is transduction? What is lysogenic conversion? In transduction, phage is acting only a vehicle. Zomato. Pick, drop. That's it. But in lysogenic conversion, phage का own DNA is acting as a new genetic element. So please understand the wordings. Okay? The last one, the fourth one. Till now we have seen three. We have seen what is transformation. We have seen what is transduction. We have seen what is lysogenic conversion. Now last I am coming conjugation. The last one I am coming is the conjugation. Conjugation is sex in bacteria. Sexduction. It is also known as sexduction. Sexduction. Okay? That is mating in bacteria. Physical contact. So in transformation, transduction, lysogenic conversion, the bacteria were not coming physically closer. Here the two bacteria come physically closer. Can you see this is one bacteria? This is another bacteria. They are coming physically in contact with each other and they make a tube between them. This tube is known as conjugation tube. This tube is known as which tube? Conjugation tube. This tube is conjugation tube and it is made by Fmge. Have I taught you what is Fmge or pili? It is an organ of adhesion. ये organ of adhesion जो होता है ना Fmge, that makes a tube. This is known as conjugation tube or sex tube, via which the plasmid of one bacteria get replicated. One copy is kept by himself and one is given to the partner. हमेशा पूरा partner को नहीं देगा. One copy kept by himself, one is given to the partner. Got it? है ना? So this is known as conjugation. Conjugation is of three types that I will explain you now. Conjugation is of three types. Can I explain the conjugation? फिर मैं पूरा summary देती हूँ. So conjugation is of three types. The first is the F plasmid, F factor, sex factor या fertility factor. Second is cold, third is R. पहले F वाला लेते हैं. F factor. देखो बैक्टीरियाज में भी मेल फीमेल होता है. जैसे ह्यूमंस में we have male female ना, bacteria also have male female. Can you see the two bacteria in front of you? The first bacteria have F plasmid and the second bacteria do not have F plasmid. है ना? So this is F positive and this one is F negative. ठीक है? The F plasmid is the male bacteria जिसमें है and the bacteria which do not have F plasmid is female. ठीक है? So male and female are coming closer, physically closer. They are making a tube between them. They are doing the sexual intercourse. They are making a tube between them. The male bacteria which is having F plasmid, it is replicating the F plasmid. You can see the tube. It is replicating the F plasmid. One copy is kept by himself and one is given to the partner. So at the end, both become F positive. At the end, both become F positive. In other words, if I say at the end, both become male. So at the starting, one is male, one is female. At the end of the sexual intercourse, both become male. So this happens in bacteria only. है ना? When they start, one is male, one is female. When they end, both become male. So this is the sexduction. It is known as F factor, sex factor, fertility factor. So who are F positive? Say they are males, they are donors. And who are F negative? They are females, they are recipients. Say yes, people say yes. Got it? So whenever we started, both are F positive. Can you see? This is, I'm sorry, one is F positive, one is F negative. But whenever we end, both are F positive. Okay? Yes. Okay. So this is F plasmid. Second is coal is not important. इसमें coal-like substance होता है. The third is important, R factor. R factor, R for resistance. R for resistance, I already taught you this in TDR. I told you two type of drug resistance, the MDR, TDR. मैं TDR पढ़ा चुकी हूँ. यहाँ पे revision करा रही हूँ. देखो, these are the two friend bacteria. दोनों best friends हैं. Both of them are best friends. See what is the difference between them? Both of them are bacteria. Both of them have nucleus. I'm not interested in nucleus. This one have R plasmid, R positive. This one don't have R plasmid. This is R negative. I know. Nucleus is present in both of them. But one is R positive, one is R negative. So R contain the gene for tetracycline, sulfonylurea, cephalosporin, है ना? Sulfonamides जो भी हैं, multiple. So whenever any of these antibiotic is coming here, whether tetracycline, sulfonylurea, cephalosporin, है ना? जो भी, so this bacteria is not dying because it is having the gene for that. But the other bacteria, when the same antibiotics are coming here, this bacteria will die because it don't have the gene. So the second bacteria is saying to the first, you are my best friend, है ना? We live together, and when the tetracycline will come, you will survive, I will die. It is not a good thing, ना? तू तो जिएगा, मैं तो मर जाऊंगा, और जन्म जन्म का साथ है. So you should do something for me, है ना? So give the plasmid to me also, so I can also survive. So the two bacteria are coming closer. They are making a tube between them. है ना? The first bacteria is replicating the plasmid. One copy is kept by himself and one is given to the partner. So whenever they started, one is R positive, one is R negative. Whenever they ended, both become R positive. Whenever they ended, both become R positive. Say yes. So this is R plasmid, R for resistance. Whenever they started, one is R positive, one is R negative. Whenever they ended, this is R, both become R positive. This is known as TDR. I already taught you now. Transferable drug resistance. For multiple drugs. You can see it is for multiple drugs, not one drug. At a time, multiple drugs are there. Say yes. You are marking everything in the notes. मैंने notes में सब लिख के दिया है ताकि तुम्हें कुछ भी ना लिखना पड़े. Everything is given in the notes. You just mark it, highlight it. है ना? एक और एडवाइस है, आज प्रिंट आउट नहीं लिया तो कल लेके बैठना फॉर द नेक्स्ट 3 days. एटलीस्ट नेक्स्ट 3 days अगर प्रिंट आउट हाथ में रहेगा, इट विल बी वेरी इजी फॉर यू. डे 1 का ठीक है, कोई बात नहीं. डे 2, 3, 4 का प्लीज टेक अ प्रिंट आउट फ्रॉम टुमारो. प्लीज टेक अ प्रिंट आउट. टुडे इवनिंग आफ्टर द क्लास टेक अ प्रिंट आउट. है ना? इट विल बी इजी फॉर यू, आई गेस, बट इट्स योर चॉइस. ठीक है? ओके. But you are asking, Bilal, permit bacteria to evolve resistance to the antibiotic. Yes. Okay. Okay. So you can see it is for multiple. I know. So this is R factor. This is R factor. See, it is having multiple antibiotic, na? T, tetracycline, SU, sulfonylurea. In सबको कहते हैं R determinant. Multiple R determinants are there. How many R determinants? You will say several drugs at a time. It can be many drugs at a time. लेकिन one multiple R determinant तो है. One RTF है. What is RTF? Resistance transfer factor. It is known as RTF. Resistance transfer factor. It is always one. It is always one. It is required for transfer. For example, कोई बैक्टीरिया में R determinants तो multiple हैं. There are multiple R determinants. लेकिन उसके पास RTF नहीं है, तो वो खुद तो prevent हो जाएगा इन सब से, लेकिन अगले को नहीं दे पाएगा. It cannot transfer it to that partner. For transfer, RTF is required. My point is that, है ना? So RTF is always one, and it is required for transfer. So this is R. You got it. So we are done with the 4 ways of gene transfer. We will revise on one page. Can anyone help me? Let me draw. Let me draw. So this is 2 bacteria. This is 2 bacteria. This is only 1 bacteria. And this is again 2 bacteria. ठीक है? You will tell me now. I will not tell you. You will tell me. Here I want to draw transformation. है ना? Here I want to draw transduction. ठीक है? Here I want to draw lysogenic conversion. Lysogenic conversion, and here I will draw conjugation. My writing is little bit poor, so please forgive me for that. है ना? I cannot, अच्छा नहीं लिख पाऊंगी मैं बहुत. Anyways, so चलो, tell me transform. Okay, let me draw 1, 2, 3, 4. Let me draw the colors. Okay, so tell me how we do the gene transfer between the 4. Okay, और थोड़ा सा देख लो. Make a lines between them. No confusion. Can you help me? The 4 ways of the gene transfer. Okay, can you help me? Both 4 ways of gene transfer. Yes, what I'm saying. What is transformation? In transformation, the first bacteria is dead. So it will rupture. It will rupture. The free DNA will leak from the first bacteria and it will enter in the second bacteria. Mark the word free. Free or naked. Free or naked DNA will be there. Yes, free or naked is the highlighted word. Not only this, you have to learn the Griffith experiment to prove this. है ना? Griffith is for this, not for this, not for this, not for this. Griffith is for transformation. Griffith में the bacteria is pneumococcus, and you know the 4 mice and their fate. ठीक है? The second वाला transduction में there is a bacteriophage. Bacteriophage is pick and drop. It is taking the DNA from here and dropping it here. So it is pick and drop. That is transduction, pick and drop. The third one, lysogenic conversion, is also a bacteriophage, but it's not pick and drop. It's the phage own DNA given to the bacteria. Phage own DNA. See, I'm using the colors very judiciously. देखो मैं कौन सा कलर कहाँ कैसे बना रही हूँ. ठीक है? So the phage own DNA is given to the bacteria. The last one is the sex in the bacteria. The 2 bacteria will come closer and make a tube between them. Make a tube between them. Not only this, they will make a tube. The plasmid from one get replicated, 1 copy is kept, and 1 is given to the partner. That is conjugation. Conjugation is of 3 type or 2 type. F factor, जिसमें male female है. R factor, कोल वाला इतना important नहीं है. Yes, I'm done. Okay, yes, this is also known as sexduction. This is also known as mating or physical contact. Everyone got it? Say yes, people, respond. Appreciate my efforts. ठीक है? आगे चलें. So these are the 4 methods of gene transfer in a bacteria. है ना, ये बंद हो जाता है टेलीग्राम. ओके ओके. What is the difference between F factor and R factor? Both are plasmids, Shivam, but the properties is different, na, Shivam? So F factor is providing the maleness in the bacteria. The bacteria which have the F factor, it means a male bacteria. And R factor is providing the resistance against the antibiotics. So the genes they are carrying is different. F is carrying the gene for fertility and R is carrying the gene for the resistance. That's it. But the mechanism is same. Okay, both will be plasmid. Absolutely right. Okay, can we go ahead? Anna? So the same thing is written in front of you. In transformation, it is the free DNA. In transduction, it is the bacteriophage pick and drop service. In lysogenic conversion, it is the phage DNA giving own DNA to the host. And conjugation, it is the physical contact sextation. I hope everyone got it. You get many questions. Same thing. Hannah? Now the next thing is the blotting technique. The next thing in the genetics after this is the blotting technique. What do you mean by blotting technique? Listen, there are many microorganisms in this world. Microbiology is all about microorganisms, right? In the microorganisms, we have bacteria, viruses, fungus, and parasite. Right? All human beings have a unique DNA. You believe? Unique DNA means unique fingerprint. We all have unique fingerprint now. Our fingerprint do not match with each other. So in the same way, all the organisms have a unique gene or unique DNA, which do not present in any other. Unique gene. From the DNA, RNA is made. So unique RNA is also there. From the RNA, protein is made. So unique protein. So all the organisms have a unique DNA, unique RNA, and unique protein. So if we can detect it, we can make the diagnosis. Hana? Just suppose I'm having a specimen. Hana? Just suppose I'm having a patient in my clinic. Just suppose I'm having a patient in my clinic. This is my patient. Okay, this is my patient. This is my patient in my clinic. My patient is having certain symptoms. Based on the symptom, I'm having suspicion that my patient is having this disease. For example, I'm having suspicion my patient is having syphilis or HIV or any disease, any disease, XYZ, any disease. Based on my symptoms, this is my suspicion. I'm not sure. I'm not sure. I want to confirm it. So what I will do, I will take a sample. I will take from the patient, I will take a sample. Now, whatever is my suspicion, I'm having 2-3 differential. Maybe it is syphilis, maybe it is Haemophilus ducrei, maybe it is this. I'm having multiple. So whatever organism they cause, I know the unique DNA of each of them. So in this specimen, I can check कि ये जो unique DNA है वो किसका है, इसका कि इसका किसका, या फिर unique RNA किसका है, या तो unique protein किसका है. इसे कहते हैं blotting technique. What it is known as blotting. So my point is that blotting technique. My point is that in the blotting technique, we can test DNA, we can test RNA, we can test protein. There are 3 types of blotting. We can test in a sample. Say yes, huh? So to test, don't go in the details of the procedure. Just learn there are 3 types of blots: southern blot, northern blot, western blot. The 3 types of blots are southern blot, northern blot, and western blot. Southern blot is to detect the DNA. Northern blot is to detect the RNA. And western blot is to detect the protein. ठीक है? How to learn that? और इनको ये नाम क्यों दिया? डायरेक्शंस वाले नाम हमने क्यों दिए? सदन इज अ रियल साइंटिस्ट जिसने डिस्कवर किया था. उसका नाम था E.M. सदन. E.M. सदन वाज अ रियल साइंटिस्ट जिसने डीएनए डिटेक्ट किया था. बाद में आरएनए डिटेक्ट किया तो उसी से सदन से हमने नदन बनाया, नदन से वेस्टर्न बनाया. उसको ऑनर करते हुए या मजाक बनाते हुए, आई डोंट नो, बट सदन वाज अ साइंटिस्ट जिसने पहली बार डिस्कवर किया था. ठीक है? सो आई एम हैविंग अ निमोनिक फॉर यू. साउथ में डोसा इज वेरी फेमस ना. साउथ इंडिया में पीपल ईट डोसा वेरी फ्रिक्वेंटली, है ना. इट्स वेरी फेमस. सो डी फॉर डोसा, डी फॉर डीएनए, है ना. सो सदर्न इज फॉर डीएनए. नॉर्थ में रोटी इज वेरी फेमस. आर फॉर रोटी, आर फॉर आरएनए. सो नॉर्थ की रोटी, नॉर्थ नदन इज फॉर आरएनए. एंड वेस्ट में पिज्जा इज वेरी फेमस. पी फॉर पिज्जा, पी फॉर प्रोटीन. सो वेस्ट का पिज्जा. वेस्टर्न ब्लॉट इज प्रोटीन, है ना. सो यू टेल मी साउथ, नॉर्थ एंड वेस्ट. आई मीन टू से सदर्न, नदन एंड वेस्टर्न. यू टेल मी साउथ में क्या फेमस है? डोसा. डोसा मतलब डीएनए. नॉर्थ में क्या फेमस है? रोटी. रोटी मतलब आरएनए. और वेस्ट में क्या फेमस है? पिज्जा. पिज्जा मतलब प्रोटीन. So you will never forget. You get questions on that. Say yes. Say yes. Everyone got it? So please don't get confused and you get questions on that. Please learn that. This is the mnemonic. The last thing here in the genetics, after that we take a lunch break. 5 minutes more. Okay? Just 2-3 minutes more. The last thing here we will see the PCR. What is polymerase chain reaction? What is PCR? Polymerase chain reaction. So let me come on the last topic here. Polymerase chain reaction. PCR. What is PCR? नाम में ही है. It's a chain reaction. It's a chain reaction. It was discovered by a scientist, Carey Mullis, and he got the Nobel Prize for that. It was discovered by a scientist, Gary Mullis, and he got a Nobel Prize for that. What actually we are doing? Just suppose I'm having a sample. In the sample, any microorganism is there. This is the microorganism. It can be bacteria, it can be virus, it can be fungus, it can be parasite. But the point is that it's very less in quantity. It's very less. The quantity is 10 to the power -9 or -12 milligram. बहुत कम है, ना के बराबर. It's very minute. So I cannot test it. I cannot test it. Which bacteria, which virus. So I want to do the blotting, but the sample is very less, very minute. I want to amplify it. I'm using the word amplification. I want to amplify. Amplified. You know what is amplification? Making multiple copies. So out of few copies, I want to make million of copies. Million copies in a fraction of time, within few minutes. Is it possible? It is possible by PCR. Make a chain reaction. Chain reaction सुना है? Chain reaction से within few minutes you can make million of copies. है ना? This is known as PCR. Got my point? So it amplifies the DNA. है ना? DNA or particular gene you want into multiple copies. Large amount of DNA within a fraction of time. So how do we do that? There is a machine known as PCR machine or thermal cycler. Can you see? This is a machine. We have a machine. This is known as PCR machine or thermal cycler is there. Got my point? PCR machine or thermal cycler we all have. Okay, say yes. So we have PCR machine or thermal cycler. So it is all automated. We just put our sample inside that. इसके अंदर अपना sample डाल देना है ना? जैसे ही आप sample दबा के start का बटन दबाओगे, you start, है ना? So the temperature become 95 inside this. Automatically it will become 95. At 95, the double-stranded DNA will split into single-stranded DNA. अब तुम्हारे सैंपल के अंदर कोई तो माइक्रो ऑर्गेनिज्म होगा ना. द सैंपल कंटेन आइदर द बैक्टीरिया और द वायरस और द फंगस और द पैरासाइट. व्हाटएवर ऑर्गेनिज्म योर सैंपल हैव, the DNA of that organism will denature. It is known as denaturation. This step is known as denaturation, in which the double-stranded will break to single-stranded. Can you see? The double-stranded will break to single-stranded. You can see a break in between. The double-stranded DNA break to single-stranded at 95 degrees Celsius. That is known as denaturation. ठीक है? Automatically the temperature will become 55 after that. You don't have to do anything. First temperature become 95 and within a fraction of second, एक second भी ज़्यादा है, within a fraction of millisecond, automatically temperature 55 हो जाएगा. So whenever 55 is occur ना, तो one is leading strand, one is lagging. I guess you may be knowing leading क्या होता है, lagging क्या होता है. One is leading, one is lagging. दोनों पे आके primer लग जाएगा. So primer will attach at both the strand. है ना? This step is known as annealing. Annealing means attachment of primer. It occurs at 55. It occurs at 55. Annealing means attachment of primer. Primers already इसके अंदर डले हुए हैं. Primers are already installed inside it. Primer will attach on the leading and lagging. As soon as the primer will join, again the temperature automatically becomes 72. Automatically, you don't have to do anything. Everything is automated. You don't have to press the button कि अब 95, 55 अपने आप होगा वो. It is automated software which is installed inside that. है ना? So the temperature becomes 72 automatically. है ना? So when the temperature becomes 72, primer is already there ना. Now it will extend. You know, adenine form bond with the thymidine and guanine form bond with the cytidine. You know na. So these all four nucleotides are adenine, guanine, cytosine, and thymidine. So they will form bonds and corresponding bonds are formed. So from 2, we started with 2, है ना? We are ending with 4. हमने 2 chains से start किया. We are ending with 4. This is known as extension. So 3 steps are there. Can you tell me the 3 steps? क्या हैं 3 steps? Denaturation, annealing, extension. बोलो एक बार. Denaturation takes place at 95. Annealing takes place at 55. Extension takes place, I forgot to write, it's 72. Extension takes place at 72. And automatically, first this, then this, then this. है ना? So we started with 1 and 1, 2 strands of DNA. Whenever we ended, they become 4. Now listen. Again, temperature become 95. So the product of the first cycle become the reactant for the second. मेरी बात सुनी क्या? The product of the first cycle become the reactant substrate for the second cycle. So these 4, ये 4 की 4 will become denatured now. है ना? Again annealing, again extension. So 4 में से बन जाएंगे 8. Again 8 में से 16. The cycle continues. We start with 2. You can see. We start with 2. We end with 4. 4 to 8. 8 to 16. The cycle continues. Never ending cycle. So the 3 temperature, first 95, then 55, then 72. Then again 95, then again 55, then again 72. Then again 95, then again 55, then again 72. You got my point? So at 95, there is denaturation. At 55, there is annealing. At 72, it's extension. Got my point? Got my point? So this cycle continues, 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 and in this way we start with one सिंगल स्ट्रैंडेड डीएनए है ना, और डबल स्ट्रैंडेड डीएनए मैंने 2 कॉपीज से स्टार्ट किया, 2 से पहले 4 बन गई है ना, 4 से बन जाएंगी 8, likewise it will continue. How many of you got it? How many of you got it? First tell me. I will repeat. First you tell me you got it. How many of you got it? The 3 steps. You have to learn the 3 steps in PCR. First is denaturation, then annealing, then extension. Tell me the temperature of the 3. Denaturation takes place at 95, annealing takes place at 55, extension takes place at 72, and they repeat. The product of the first is the reactant of the second. Okay, anyone have any confusion? PCR, polymerase chain reaction. I was having a small video, but I'm not finding it right now. I forgot to put it here. It was a small GIF in which the 3 steps are very clear to you. So maybe after the lunch, I will put that GIF and you will have a more clearing there. Okay, yes. ठीक है, ये 55, ये 72. Yes, ये तो लिखा है. Yes, the 3 temperatures. Okay, so same thing is written to you. So read the question, tell me the answer. Read the question. Yes, it's repeated again and again. DNA is detected by which blot? Southern, western, northern or eastern? DNA for dosa. Dosa कहाँ मिलता है? So it is southern. You all are right. Dosa is always southern. Dosa is always southern. Very good, very good. है ना? So if I change the question from DNA to RNA, you know answer will become northern. And if I change it to protein, answer will become western. So you know the answers. I'm not changing it. Tell me the name of the bacteria which is used in Griffith experiment. Tell me the name of the bacteria which is present in Griffith experiment. Huh? So of course, the correct answer is Streptococcus pneumoniae. है ना? Okay. Griffith experiment is to prove transformation. It is used to prove transformation. ये भी एक PYQ है. Right? The next question. Movement of DNA from one bacteria to another through a tube, a connecting tube, or through a pilus. Is it transformation, transduction, conjugation, or lysogenic conversion? The movement of bacteria, DNA, from one bacteria to another through a tube. Yes. So they are talking about the physical contact between the bacteria. They are asking for conjugation. So correct answer is conjugation. If I change the question, है ना, through tube की जगह through bacteriophage कर दूं, तो what is the answer now? Through bacteriophage कर दूं तो what is the answer now? Through bacteriophage. Yes. In that case, answer will become very good. In that case, answer will become transduction. ठीक है, ट्रांसडक्शन, वेरी गुड. और उसको भी चेंज करके अगर मैं फ्री डीएनए कर दूं तो आंसर विल बिकम ट्रांसफॉर्मेशन. ओके, सो यू गेट क्वेश्चन ऑन ऑल थ्री. Got it, huh? So you want to continue with the sterilization or you want a lunch break now? What do you want? You want to continue or you want a lunch break now? So still we are pending with one chapter here, sterilization, number 1, sterilization, and after that we have complete immunology. We have immunology. Complete immunology. These 2 things are pending. From general only sterilization is there. So a big chunk is completed. And immunology is small. It's not very big. So you want lunch break? Most of the students want the lunch break, right? So we will take a lunch break. Okay. So we will take 1 hour lunch break. I will be back at 3:15 say. Okay. Till that there is lunch break. After that we will continue with sterilization. And after that we will continue with this thing. What do you say? Immunology. Okay, Ji? So let's see after the lunch. Okay, I'm ending this and a new link will be provided to you. Okay, so be on time, eat something light and be energetic after the lunch also. After the lunch, we will continue maybe till 5:30, maybe 6. Let's see. We will continue. Okay, bye-bye. Okay, next 3 hours. Okay. I'm ending it. Bye-bye.